Hello, class, and welcome to Chapter 12. Chapter 12 is going to go over genetics, mainly at the human level, but also going to be focusing on other organisms as well. So we start out the chapter by looking at heredity, mainly looking at Gregor Mendel's pea plant experiments. And then we go into some of the interpretations of his results that have led to the first law and second law of heredity. But then we're going to go into some of the complicating factors. While Mendel's work really did help us understand genetics for the first time, we have learned that over studying human genetics that there are a lot of different complicating factors, and we'll go through each ones of those, and we'll talk about gene disorders from a recessive and a dominant stance. So, mystery of heredity. So as we see here, before the 20th century, there were only two concepts for the basis for all ideas of heredity. One is constancy of the species, which means that people used to believe that others could not cross, animals couldn't cross with each other, which is more or less true. However, we can go out into the French Quarter and we can see the mules. These are crosses between a horse and a donkey, and there are several other examples of some species in nature that can cross with each other. However, during this time, during the Middle Ages, most species were looked upon as being fixed and unchanged because most people didn't live very long, so they didn't see some of these changes occur over a period of 80, 90 years, which is now the typical lifespan. These people were only living to be 20, 30 years old. The other thing is direct transmission of traits, and this got a lot of people killed, essentially, especially during the witch trials and other different instances like that along our history, where people believed that traits were passed on directly to offspring and were blended between the mother and the father. So therefore, as I see it show you here, if the mother had red hair and the father had brown hair, the expectation would be that the offspring would have reddish brown hair. The reality is, genetics shows us that there are several recessive genes, as we'll talk about throughout the lecture. And these recessive genes can pop up periodically, and so sometimes you can have people with brown hair having a red-headed child, and this is due to the recessive gene. So the paradox that became in this particular instance is, if blending occurs, why don't all individuals eventually look alike over time? And so this was a big blow in the hole to this belief. However, during this time, some women were actually hung, stoned to death, any other sort of terrible outcome that resulted in their death because of sometimes what the child looks like, and they assumed that the woman was cheating on the man. So some of the early work before Gregor Mendel came from these two gentlemen, Joseph and T.A. Joseph was looking at tobacco strains, the Bruce hybrids, that differed from both parents, and so what he was looking for was a better tobacco plant, right? And so during this time, the tobacco is still a very cash positive crop. During Joseph Coulter's time, he was looking for ways that the tobacco plants could be more hardy, produce bigger leaves, and so he was playing around with crossing different strains. And so he's considered a father of hybridization, even though we really didn't show too much from his work because he didn't write it down that well. The other one is T.A. Knight. He was looking at garden peas, much like we're going to see in the work that Gregor Mendel was doing. And what he was showing here is that when you cross two species of garden pea, you end up with one strand, the first generation, which we can think of as like the children of those two strains, resembling only one of the parents. However, when we got to the second generation, which would be like the grandchildren, they had different qualities of both. So he was the first to notice this, which Gregor Mendel did also. The only problem is that he didn't quantify anything, so he didn't write it down or actually put numbers next to his experiments. So he's more or less kind of forgotten into the archives of science. However, Gregor Mendel, one thing he did well was take notes. Gregor Mendel was an Austrian who was educated in a monastery, which wasn't uncommon at those times. He really wanted to become a science teacher, but unfortunately he didn't pass the teacher's required exam of the time. And this is one of those particular exams that you're only allowed to take once. And so once he didn't pass it, his dreams of being a scientist or a teacher were pretty much destroyed. So he went and spent the rest of his life in a monastery, and eventually working his way up to an abbot. But what he was really interested in was still the science, and so he started studying peas in the garden of the monastery, mainly because peas were very small. They were easy to grow. They didn't spend a lot of they didn't take a lot of space up. And so he had also known about T.A. Knight's work with the peas and knew that different pea hybrids could be produced. And another key is step 4, which is that peas can self-fertilize, and they can also be cross-fertilized by a different strain of the pea plant. And this is not uncommon within plants where they can self-fertilize or be cross-fertilized. So he had 3 stages to his experiments. One was to produce a true breeding strain. So a true breeding strain is one that every time we plant that seed, we know what that plant's going to look like, whether it be purple flowers or white flowers or green seeds or yellow seeds. Every time we plant that particular seed, we know what it's going to produce. So that's a true breeding strain. And this is going to be different from what we're going to see later when it comes to a heterozygous strain. And so, keep that in mind. This true breeding is going to come up a few times. Cross-fertilized true breeding strains have alternate forms of a trait. So in this case, what they're showing is that when we cross a purple flowered plant with a white flowered plant, usually what we get in return is all purple. And so here we cross-fertilize 2 true breeding strains, one being a purple flower, the other one being a white flower. And we're going to see that after that cross, they all look like the purple plant. We're going to look at the genetics and see why that is. He also allowed the hybrid offspring to self-fertilize. So imagine this: you cross a purple and a white, you get whatever you get. From that, every generation past that original cross is just self-fertilized. So there's no chance that new genetics are going to be inserted into that particular plant, which is important for his experiments. So we call what he was doing mainly a monohybrid cross. So mono meaning one, hybrid, we're looking at only one difference between the 2 plants at a time. So here cross studies only 2 variations, either purple or white, as I've been reiterating, or it could be something like green and yellow seeds, wrinkled or smooth seeds. There's several different things he could have been looking at, but he only looks at 2 variations of a single trait. There's no such thing as yellow, red, orange flowers for this particular species. So this is really, really simple and is going to lead us down the road to the problems that we see in humans, that this is deceptively simple. Only having 2 possible outcomes is very different from all the multiple outcomes that we could potentially see in human genetics. However, it can lead to important conclusions about the nature of inheritance. So again, we're going to keep reiterating that while Mendel's work was incredibly important for starting our understanding of genetics, his experiments were more or less along the most simplistic, and some would say that Mendel was very lucky that the particular plants that he was looking at only had 2 variations of these single traits. He looked at a total of 7 different traits. Each one of them only had 2 possible outcomes. So when we look at the way that his experiments were set up, we're going to call them F1 and F2. F1 is going to be the children, or the first generation, while the F2s are going to be the second generation or grandchildren. The F stands for filial, which essentially translate in Latin to son. And so, again, first generation being the son or the first children. So these are the offspring produced by crossing the first 2 true breeding strains. So imagine this: you got your purple plant, you got your white flowered plant, you cross them together because they are 2 true breeding strains. Every time we plant that purple flowered seeds, we get purple flowers. Every time we plant the white flowered seeds, we get white flowers. And so when he crosses those, the very first generation he calls the F1. What's interesting is that, the F1 plants all resemble only one of the parents, and I'll give it away, they actually represent the purple plant parents. This is referred to as a trait as dominant. So the ones that come out in the very beginning are the dominant trait. The other trait, in this case the white flowers that none of them look like, is the alternative trait, which is recessive. So these are 2 key vocabulary terms, dominant and recessive. The F2 generation, or the grandchildren, were the offsprings resulting from self-fertilization of the F1 plants. So as you can see here, the purple and white plants, both of those being true breeders, were crossed. And what we'd see in the F1 generation is that they would all be purple. However, in the F2 generation, what we'd see is a ratio of roughly 3-to-1. And this is what made Mendel's work stand out, is that he quantified things. He actually wrote down the numbers and gave us solid statistics that are necessary for us to actually come up with a conclusion when it comes to scientific experiments. So here's some of the other things he looked at: yellow versus green. When he crossed them, eventually the F2 generation will come out with a 3-to-1 ratio of yellow to green. So green would be the recessive, yellow would be the dominant, with the purple and white flowers. Also the purple is dominant, the white is recessive. Seed texture, round or wrinkled, pod color, green or yellow. Again, all these things came out, and notice how the ratios are all right at or almost on top of 3-to-1. And so that means that for every 3 purple plants in the F2 generation, we'd get 1 white. So when we actually look at it, the 3-to-1 is truly a 1-to-1 ratio. And so follow along what's happening here. During Mendel's time, he had no idea what DNA was. He didn't know what genetics were. And so for him, all he was doing was looking at the colors of the flowers. And so when he looked at them, he seen 3 purple flowers and only 1 white. And so his ratio came out to 3-to-1. What he really had found, which he wasn't aware of, is that he had gotten one of the purple plants was back being true breeding. That means both its genes. We have to remember that we have 2 copies of our genes, right? We have one from mom and one from dad. In this case, the true breeding purple that you see there on the far left at the bottom is one that has 2 copies of the purple flower gene, essentially. That means there's no chance that it has any capability of making a white flowered plant. So here the 2 true breeding plants are going to be the purple one on the far left, which is 1 of the 1-to-1 ratio. The white one on the far right is another true breeding that is always going to produce just white flowers. That means it has 2 copies of the white flower gene and does not possess the capability of producing a purple flowered plant. And that's the 1 of the 1-to-1 on the right hand side. Now the 2 comes from the 2 middle purple dominant plants, which are called non-true breeding. That means it has 1 copy of the purple flower gene and it has 1 copy of the white flower gene. We also call these heterozygous versus homozygous. Homozygous means that they have 2 copies of the same gene. Heterozygous means that they have 1 copy of the gene that might be, for in this case, purple flower. The other copy is white. But since the purple flower gene is dominant, that means that all we're ever going to see, in the most part, is going to be the purple flowers. However, on occasion, the 2 white genes will show up and give us a white flowered plant out of these, and thus they're not true breeding because we're not sure what we're going to get at any given moment. So we call them not true breeding plants. So from his work, we were able to show that plants did not have any intermediate traits, meaning that we didn't get something in between purple and white. We didn't get a lavender colored plant. It was very intact and discrete, meaning you were either purple flower or white flower. For each pair, there was 1 trait that was dominant. So in this case, the purple flowers are dominant, the yellow seeds are dominant. We can go back and look at that table that had all the different dominant versus recessive. Pairs of alternate traits examined were segregated among the progeny of a particular cross. So in this case, he would set them aside and started looking at them individually to ensure that he got his numbers. Even if he didn't know what he was doing at the time, his strategy really helped us geneticists and scientists down the road since his work to truly understand what he's seen. And so what we truly found out was the F2 generation, remember this is the grandchildren, had a ratio of 3-to-1. And again, when we look at it genetically, it's truly 1-to-1. All right, so here's a bunch of vocabulary terms. Many of them we've already hit on, but I want you to definitely take notice of this page. So parents transmit discrete factors. Well, it's in parentheses. Or it's uh, let me start over again. Parents transmit discrete factors, and the reason why that's actually got the parentheses around them is that this is what we call genes now, but in his actual lab manuals, he called them factors. And again, genetics wasn't understood, DNA wasn't discovered yet, and so this is truly just looking at how flowers and the pea plants grew and taking notes of them. Each individual receives one copy of gene from each parent. This is something that's true, right? You know, we have one copy of our chromosomes that's from mom and one that's from dad. Not all copies of a gene are identical. So when we look at an individual gene, again, 3.2 roughly billion nucleotides translates into roughly about 20,000 genes in our chromosome. Of those, you're going to have some that are alternative forms of a gene. So remember, you get one copy from mom, one copy from dad. So you got 20,000 genes from mom, 20,000 from dad. And so if you have one particular gene that's coding for two different things, so for instance, maybe mom gave you the gene for blue eye color and dad gave you the gene for brown eye color, those are what we call alleles. So if they are alternative forms of a gene, we call them alleles. Same thing can be with the flower color, pea color, whatever it might be. Those are different variations of a gene. Now, some genes only code for a particular enzyme, and everybody's got that same exact allele. But some things like eye color, hair color, that can be many different alternatives of an allele. It's not just going to be two different possibilities or even one. Now, if you have the same gene from both mom and dad, we call that homozygous. And if we have different alleles for a particular gene, we call that heterozygous. So again, some key terms there. Alleles remain discrete, so therefore there is no blending like the people originally believed. Essentially, the genes tell exactly what needs to be done. They don't mix together, at least in Mendel's work. Presence of allele does not guarantee expression. So in this case, if you have the recessive allele, but you also have the dominant one, the recessive is going to stay hidden. And so you'll never have that, that particular trait being expressed. And so we call that our phenotype. Our phenotype is what we actually look like, our physical appearance, or what the pea plants actually look like, or whatever we might be studying genetically. We're looking at what the actual appearance is. That's our phenotype. However, now that we have the capacity to go and do DNA sequencing, we can also look at the genotype. And the genotype is a total set of alleles in an individual, whether it be a plant or an animal or a human. This is the total set. So both mom and dad's genes, looking at the recessive genes, even though they may never express, we can still see if you have the potential or you are actually carrying those particular genes, which becomes important nowadays for looking at different diseases and possible, and being able to predict a person's chances of having a particular disease by looking at some of these recessive genes that we didn't have access to before DNA sequencing. So we look at the principle of segregation. This is also known as the first law of heredity. And here we have in quotes, the two alleles for a gene segregate during gamete formation or are rejoined at random, one for each parent during fertilization. This requires you to go back and look at chapter 11 and looking at how gametes actually form. We know for sure that we have the chiasmata connection as well as the homologous recombination events that occur where genes are bounced around between the two different chromosomes. And so this is basically what the first law of heredity is saying, is that the genes segregate during gamete formation. This is the process of making the sperm or the egg, and are rejoined at random. Again, as far as we are aware from all the studies that have been thus far on homologous recombination, it is truly a random event, that there isn't any sequence of events that happen to cause a gene to bounce from one place to another, at least with our current understanding. Now, physical basis for allele segregation is the behavior of the chromosomes during meiosis. Yet again, homologous recombination, and the fact that You just have to go back and look at chapter 11 and brush up on exactly what's happening. Mendel had no knowledge of chromosomes or meiosis because they hadn't been described or discovered yet. So then we get into the Punnett squares. This is named after Reginald Punnett. What he was looking at was a way to come up with a prediction on the possibility of what the plants may look like. And so what he came up with was a novel shorthand for dominant and recessive alleles. Dominant is always given a capital letter. In this case, he's looking at the purple and white flowers, and so the purple flowers got a capital P. The recessive allele gets a lowercase p. Now, you can use whatever letters you want. In this case, it just so happened P was for purple, and so that's why it was being used. So a true breeding white flowered plant, remind yourself of what true breeding means. True breeding means that it has a copy, the two copies of the same gene are exactly the same. So in this case, for it to be a white flowered plant, it's going to have two lowercase p's, signifying that both genes for flower color are white. This also means it's homozygous recessive. So that word should be able to instantly bounce out and say homozygous, meaning that both genes are the same. Recessive, suggesting that both of the genes are the recessive version of that trait. Now, the true breeding purple flowered plants are going to be the exact opposite. They're also going to be homozygous dominant, but they're going to have two uppercase P's to symbolize that. And then a heterozygous purple flowered plant is one that has a copy of the purple gene, which is signified there with the uppercase P, and it has one copy of the white flower gene, which is lowercase p. So this makes it heterozygous. It means it has one of each, and yet because it has the dominant gene there with the purple flower, that means all the flowers are going to be purple in this case. So we can look here at some of the Punnett squares. I want to focus mainly on the one on the right-hand side with the purple and white. What we're seeing there is a very simple grid that's set up. You have on one side of the grid, you have the two genes for the purple parent. So there's a capital P in row 1, and there's a capital P in row 2. And then we look at the top, there is the lowercase p symbolizing the white flowered plant, and so column 1 has a lowercase p, as well as column 2 has a lowercase p. And all you got to do is just bring them down. So the lowercase p comes down into each of the boxes. The uppercase P goes across into each of the boxes, and so this is going to be the F1 generation. Now remember, what Mendel found was in the F1 generation, even when he crossed these two plants, they all came out purple. And what we can see here is in the Punnett square, all four possibilities are all purple flowers, and that's because all four possibilities are heterozygous. Notice every single box is exactly the same. Now, remember what he did next is he allowed the F1 generation to self-fertilize. That means there's no chance that they're going to get any sort of new genes being introduced. And so when he does that, we're going to have a Punnett square that looks like the bottom, and that's going to be an uppercase P in the first column, a lowercase p in the second column, and the same thing in the rows 1 and 2. And so when we bring those across, this is where we get that 3-to-1 ratio that actually is 1-to-2-to-1. What we have there in the top left box is a purple flowered plant that has both copies being dominant, so that would be a true breeding plant. The bottom right is a true breeding recessive plant because it has both copies being the recessive gene. Now we still end up with two plants that are heterozygous, one there in the top right and one there in the bottom left. Both of those are heterozygous because they have one copy each of the gene. And so that's where the 1-to-2-to-1 ratio comes from. We have one plant that is dominant, dominant homozygous. We have one that is recessive homozygous, and we have two heterozygous, so 1-to-1. Now, when we look at human traits, it's very difficult to find many traits in the human genome that are actually controlled by a single gene. What we're going to see is that There are a couple. So in this case, there is the dominant pedigree that we see here on the right. And what we're looking at here is juvenile glaucoma. And so this is a particular trait that we were lucky enough to have a French family that kept a very meticulous log of all the family members dating all the way back to 1495. And they actually write down the particular problems that these particular people had. And so we're able to track their pedigree. And pedigree just means being able to track particular genes through many different generations. And so here on the right-hand side, we have males being represented as squares and females represented as circles. You have the ones that have the gene, again, being a dominant pedigree. That means you only need one copy of the gene to have this problem of juvenile glaucoma. So in this case, the very first generation, it appears from this particular pedigree that the male was carrying the gene. Gets married and has 3 children. Of those 3 children, 2 of them have the same affliction. One of them doesn't get married, which is there on the far left, number 1. You know, the male there just stays, doesn't have children. The other 2 end up getting married or having children in some fashion. And what we see there is that 2 of the children that had the dominant gene married people that did not have the dominant gene, but yet all 3 of their children all were carrying the dominant gene for juvenile glaucoma. And so this shows you the power of a dominant trait. Now, a recessive trait is much more difficult to follow because you end up having to have both copies in order for the recessive trait to show itself. So sometimes it's difficult to track knowing which of the parents actually was carrying the recessive trait because their phenotype, or again, remember phenotype means what did they look like, their phenotype is not going to be expressing that recessive gene unless they have both copies. So here when we look at first generation, we're looking at a particular recessive pedigree, which is albinism, which is the lack of pigmentation in the skin. And so this would be somebody we refer to as an albino, or an organism such as an albino snake or alligator. So here, generation 1, up at the top, we're not sure which one of these people, but one of them had to be recessive. And so when we look, they ended up having 3 different children. One of them, again, did not have any children further. These 2 ended up getting married. Now, what's happening here is that we're seeing that only half of them have, they have half recessive, half dominant, which is why it's shaded green and white. It just so happened out of, dumb luck that they ended up having children with a person that also was carrying the recessive gene. Now, the likelihood of these people having a child that has both recessive copies is, 25% chance, right? 3 to 1. So 75% chance, 25% chance that they're going to be homozygous recessive, which is what number 1 and number 3 was. We have 2 chances of them being heterozygous, which is right here. And we have another chance, another 25% chance for them to be homozygous dominant. So they're not carrying the albino gene anymore. So in this case, they got unlucky and they had 2 children that had the recessive trait. So they were fully expressing number 1 and number 3 would have been albinos. To look at their phenotype, what they were actually expressing. And over here, this couple, well, luckily they didn't marry somebody that was carrying the recessive gene. So all 3 of their children were not albinos. However, number 5 here is carrying the gene for it. Again, being a recessive trait, they can carry the gene without actually expressing it. Now, here's a little quirky thing, first cousins marrying, which strangely is legal in many states. But here we have that first cousins marrying. Well, this so happens to be a heterozygous female and a heterozygous male. They had 3 children. 2 of them became homozygous dominant so they're not carrying the gene anymore. And unfortunately, their 3rd child ended up being homozygous recessive and would have been an albino. So here you can see how much more difficult it is to follow recessive pedigree versus a dominant pedigree that's pretty straightforward. So some different human traits, you can look at this table. There's lots of different things on here that are referring to whether it being a recessive trait where you need to have both copies, or a dominant trait where you only need to have one copy of the particular gene. Now we can get into things that are a little bit more difficult, and again, if math isn't your strong suit, I'm not asking you to memorize how we get to these numbers, but for sure, memorize the numbers, or just understand where these numbers come from. And so remember, the very first thing that Mendel did was a monohybrid cross. This is referring to only looking at one trait at a time, whether it be plant seed color or pod color or flower color. That would be a monohybrid cross. Now, a dihybrid cross is looking at two of those traits in a single cross. So in this case, we may use a plant that has yellow seeds and also green seeds. That'd be one of the crosses, and the other cross could be round and wrinkled seeds. And so when we look at the actual seeds themselves, some of them are going to be nice and smooth spheres, and others might be a little bit more wrinkled. That would be just one of them that we're going to look at here in a second. And so now the genetics becomes more difficult. We're looking at two traits at the same time. And so when we're looking at the true breeders, we have the one there on the far left. We have true breeders that have both dominant copies of the R gene and both dominant copies of the Y gene, which is referring to smooth sphere and yellow seeds. And then we have the other one, which is a true breeding recessive trait, which is going to be wrinkled seeds, which is lowercase r's, and green, which is the lowercase y. So again, remember that being uppercase means that it's the dominant trait, lowercase is the recessive trait. So of course the F1 generation, we already know, is all going to be round and yellow, which is referred to in the next bullet here with the uppercase R and lowercase r, uppercase Y, lowercase y. And yet, so all the F1 generations, even though they're heterozygous, as we've seen before, they're still going to look round and yellow. So as he done before, he allowed the F1 generation to self-fertilize, meaning that no new genes are going to be introduced, and produced the F2 progeny, and then ultimately counted what he got there. And so when we look, it becomes another ratio of 9:3:1. And so what we're seeing here is that in the F2 generation, which is in the far right corner, you're going to see that we have 9 of them that are round and yellow. So we're going to have 1, 2, 3, 4, 5, 6, 7, 8, 9 that are round and yellow, referring to the first number in the ratio. We have 3 that are going to be heterozygous for round, but then they're going to be carried the wrinkle gene. So you see that there's 3 of them that are yellow with wrinkles. We have 3 that are going to be the opposite, where they're going to keep the round dominance, but then they're going to lose to the recessive green color. So we got 3 round and green ones there. And then we have 1 lowly over in the far bottom right corner that is going to be the only one that is carrying the recessive genes for both traits. So in this case, the green as well as the wrinkled. And so here we can see how it becomes much more difficult when we start adding in more phenotypes and start looking at the chances of any of these particular phenotypes popping up. You basically have only a 1 in 16 chance of having a plant that is both green and wrinkled seeds. So Mendel's second law of heredity is in a dihybrid cross, the alleles of each gene assort independently. That means that nothing to do with the green seed color is going to affect whether it is a round or wrinkled seed or a purple and white flowered plant. These genes act independently of each other. And again, this all occurs during meiosis, and go back and refer to chapter 11, meiosis with metaphase 1, and see how that independent segregation occurs, right? After metaphase, they split apart, and so you have different progeny going down different lines with different types of genes after meiosis is complete. So now we're just looking at probability, and this is where a lot of people make their money in terms of people that do genetic testing online or on TV like Maury, doing mainly paternity testing. but also others where they're looking at the probability or chances that maybe you know that a particular disease runs in your family, and you may be looking at likelihood that a particular child may have the same affliction. So we can start looking at probability as two different ways: rule of addition or rule of multiplication. And so the probability of two mutually exclusive events, head or tail on a coin, occurring simultaneously in the sum of their individual probabilities. So in this case, we have, when we're crossing two heterozygous, the probability of producing another heterozygous offspring is 1/2. So notice here in the picture, we cross two heterozygous, we end up with one possibility of being a homozygous dominant, another possibility of being homozygous recessive, but there's two possibilities of them being heterozygous again. So when we're crossing those, the probability of producing a heterozygous offspring is 1/2. The probability of obtaining, a heterozygous is only 1/4, plus the probability of attaining the other heterozygous is another 1/4. So again, it's just adding up. 1/4 plus 1/4 equals 1/2. And so that's the rule of addition. We can add them up and basically figure out what their probability is going to be. The other one that's much more useful and powerful is multiplication. And in this case, we're just multiplying them together. And so a probability of obtaining a lowercase, a recessive gene from dad is 1/2. Probability of attaining a recessive gene from mother is 1/2 when we go back and look at that picture again. So the probability of getting a homozygous recessive is 1/2 times 1/2. And again, if you're not good at your multiplication of fractions, that's all right. Trust me, the answer is 1/4. So the probability in this case of getting a homozygous recessive is 25%, which we can also see clearly with the Punnett square. So we can continue out these traits. So okay, we know that we have a 25% chance of getting a homozygous recessive for flower color, but then we can continue multiplying in these chances when we start adding in other factors. So in this case, if we wanted to look at something very simplistic here, it would be much more difficult than this in real life, but imagine we wanted to know if a child will have red hair, green eyes, and have a pale complexion. This becomes much more difficult because each one of those has their own probability. You have to multiply them all together, and you end up getting a really absurd small number, like 1 out of 300 or 400 chance. Only if those genes exist, between the mother and the father. What is the chances that we'll get one that has all recessive traits for every single gene we're looking at? What's the chances of them having heterozygous or having a mixture of homozygous and heterozygous? Again, this is where we can figure this all out through probability. So test cross is a way to understand if we're dealing with a homozygous dominant or recessive, or if we're dealing with a heterozygous. And so let's say we have a seed we don't know anything about. What we're going to do is we're going to plant it, because here it's a purple flowered plant, but we can't be certain, and that's what we're looking at here in the middle, a purple flowered plant. We can't be certain if it's homozygous dominant or heterozygous. So what we're going to do is we're going to plant it, and if it is a homozygous dominant, we would assume that when we cross it with a homozygous recessive, which is the key to a test cross, is that, in this case, if it was a homozygous dominant, we know that all 4 plants are going to be purple. So this is easy for us to determined by basically fertilizing it with a known homozygous recessive. Remember, this one here is not known. We're not sure which one it's going to be. Over here, the other possibility is it being a heterozygous, and when we cross it with a homozygous recessive, we're going to get some of the white flowers that pop up. Again, you can follow the Punnett squares and see how crossing a heterozygous with a homozygous recessive is going to show up that recessive trait in the offspring. This becomes a powerful tool for understanding what we're dealing with. So people that buy seeds for a living, such as farmers, they want to be certain that every time they plant that particular seed that they just purchased, it's going to come up with a known quantity. So, whether it be sugar cane or corn or whatever they might be growing, they want to know that every single one of those plants is going to essentially have the same genetics. Farmer has no interest in having one that is going to be heterozygous dominant. We want to make sure that whatever those traits are homozygous, to ensure that every time the plant is planted, we get that. So a test cross is a very powerful tool to know if we have, without actually having to get in and look at the DNA sequencing, we can basically do this macro experiment where we can see with our own eyes what the outcome is. So extensions of Mendel's work. Essentially, Mendel, when he passed away, he was pretty annoying in his monastery. And when he died, most of the people in the abbey just kind of took all his books and threw them in boxes and threw them in a closet to be forgotten. However, during renovation, that was found again, and people started looking through his books and realizing, hey, this is actually some pretty interesting work. So his work went missing for roughly about 30 years. And now when it was refound in close to 1900, we were closer to understanding what DNA was. We'll learn in chapter 14 and 15 that DNA wasn't even discovered until 1920s. So we're right there on that cusp of discovery of, hey, what are these factors? In this case, we're going to learn that they were genes. And so, unfortunately for modern genetics, where we're at currently, his model of inheritance assumes a couple things that aren't necessarily true. Each trait is only controlled by a single gene. That's not the case in most of the genetics in humans, and as well as other organisms. Each gene has only 2 alleles. Well, we can blow this out of the water by looking at all the different natural hair color types, natural eye color types, many other different traits that we can clearly see in humans. There's more than just only more than 2 possibilities. And in his pea plants, there was always a clear dominant recessive relationship, which isn't necessarily true with many genes, as we've learned over the years. So these 3 traits he got incredibly lucky with. And because of that, he was able to get some very strong data that helped us understand inheritance and help us understand how dominant recessive traits work. However, most genes that we've studied since this point don't really meet these criteria. And so what we're going to follow in the next few slides, so some of these examples of different outcomes when it comes to genetics is going to be shown here in the next few slides. So the very first one we're going to look at is what's called polygenetic inheritance. Poly meaning many. So in this case, what it's referring to is many genes that are inherited that control a particular one phenotype. And so one of the things we can look at is your height. Your height isn't controlled by any single gene. As a matter of fact, there's a lot of environmental factors that also go into your height. However, when we look at it, there are many different variations of height, right? We can have everybody from very short people, as we see here in this picture up on the top. They're basically all segregated behind signs based on their height. So you have people over here on the far left that are going to be incredibly short. You have some people over here on the far right that are incredibly tall, but for the most part, everybody kind of congregates in the middle. And that's what we see with a lot of genetics, is that we do have different variations, but for the most part, people kind of congregate, in the middle there. So the phenotype is actually an accumulation. In this case, the phenotype is your height, is accumulation of contributions by multiple genes. And so this is what we refer to as polygenetic inheritance. Pleiotropy is referring to an allele which has more than one effect on a phenotype. These are really difficult to predict and understand because it's hard to trace. One of the things that we can look at, though, is 2 particular ailments inside humans. One being cystic fibrosis, which mainly afflicts Caucasian males, and sickle cell anemia, which mainly afflicts Africans from a particular West Africa region. And so, anybody that has family roots that come from West Africa or Central Africa may be carrying a particular gene for sickle cell anemia. And so this is only one gene that controls both cystic fibrosis or sickle cell anemia. It's a single mutation that causes these problems. However, that's just one gene, but the outcome is multifaceted when we look at the phenotype from that one gene mistake. these people are going to have multiple symptoms. Somebody with sickle cell anemia is going to end up having problems with their blood flow because the particular blood cells are not shaped correctly. And this would have been a good reason for prevention of malaria, because the particular parasite that causes malaria it actually infects blood cells, but since these blood cells aren't shaped normal, these people are basically immune to malaria. So this is why we can see it traced back to these regions of Africa where malaria is of high prevalence. This would have been a natural selection process that prevented those particular people from getting afflicted with malaria, so they would essentially grow up and be able to reproduce. However, nowadays, with modern medicine, sickle cell anemia ends up being hugely problematic because we're not necessarily worried about malaria as much anymore, because there's many different medications out there that are available. So somebody that has African descent roots, but yet living in the United States or living in another first world country, the necessary need for that sickle cell anemia gene is useless. So instead, these people end up having afflictions such as strokes and heart attacks and anemia, low blood counts, unnecessarily, but yet it's only traced back to one gene. So they have all these different symptoms, but only one gene. Problem. That's pleiotropy. Basically one gene, multiple problems, multiple traits. Cystic fibrosis is a single nucleotide mutation also that prevents the cilia inside the trachea from moving. So they are not able to pull the mucus up out of their lungs, and therefore they end up with lots of different infections because the mucus ends up being a nice food source for bacteria, so they end up getting a lot of different variations of pneumonia. Sometimes they actually have to be intubated and have the mucus sucked out of the lungs for a while before antibiotics became very useful. The average lifespan of somebody with cystic fibrosis or sickle cell anemia was only, early 20s. Nowadays, these people live relatively long lives, even though they may have problems throughout their life also with these afflictions. But again, both of these are examples of pleiotropy. You have a one gene that causes multiple problems, so it makes it difficult to trace. Multiple alleles. In this case, many of what Mendel showed were only having two possibilities: purple, white, green, yellow, round, wrinkled. However, in this case, we're seeing something that his pea plants didn't do, which is actually making a blend. So here we have a F1 generation of a red and white flower. Again, these are not pea plants, so we're looking at a different plant species where they're able to look at these multiple alleles. Here you have red and white. They come together, and we're going to end up having heterozygous here. And now it actually has come out with a pink flower. So the heterozygous plants are actually going to have this intermediate trait, which we said during Mendel's time, there was no blending, but that was just particularly for his pea plants. Again, that was just something that he lucked into. Here's an example where two different alleles can blend together. And then in the F2 generation in the bottom, the two heterozygous, they're going to blend together. We're going to still get that 1-to-1 ratio as we've seen before. You get one plant that is homozygous dominant, we get that red back. You got two plants that are heterozygous, so they're still going to maintain pink, and you got one plant that is homozygous recessive, and so again we get our 1-to-1 ratio. And so this is an example of multiple alleles, and particularly it's an example of incomplete dominance. That means that even though the red flower is the dominant, we get incomplete dominance. We get something in between this pink color. Another example of multiple alleles is blood typing. So we all know that you're either A, B, or B, or O. And what are these referring to? This is referring to the sugars that can be found on the actual blood cells. So somebody that has A blood type is going to have galactosamine as a sugar on the blood. Somebody that has B is going to have galactose. Somebody that has AB is going to have both sugars, and somebody that has O has neither. So in this case, we can see codominance. Each of those genes, whether it be A or B, are both dominant. And so codominance, it refers to somebody that has AB blood type. They're showing off both galactose and galactosamine. And so this is going to be where blood typing comes from. The Rho factor is going to be what causes plus and minus. You'll learn about that more in A and P. Here we're just showing an example of a possibility that is codominant, where both A and B are going to be expressed if they're being carried in the genotype. Now, sometimes environment gets involved. And so here is an assumption that was also blown out of the water that Mendel did, which was that the environment did not affect the relationship between genotype and phenotype. We know now that your environment controls many of your genes. And so here we can look at a Siamese cat, and the particular gene that causes the pigmentation, the dark pigmentation that we see at the extremities, in the ears, in the muzzle, in the tail, and even down in some of the paw area, this is going to be turned on by an enzyme that only works when the temperature is below 30 degrees Celsius. Now keep in mind, body temperature in humans is 37 degrees Celsius. And so what we're seeing here is that when the extremities, we all know, out of our extremities end up being a little bit colder. We've all experienced cold fingertips and cold nose, cold ears. Anywhere in our extremities, it's much more difficult to keep blood flowing and keep the warmth level up. Same thing in an animal such as a cat. So the enzyme is found throughout the body. However, notice in the trunk of the animal, it's all white hair. This is because the enzyme is not turned on, because here is going to be where the animal is the most warm. We got to the extremities, we actually see that there's a darker color of the fur, and this is due to the fact that the gene is now turned on, or the enzyme I should say is now turned on because the temperature is dropped. So this is an example of how the environment can play with the genes. Very simplistic example. There are many other examples out there. However, this is a good example showing something that's very easy to see in the phenotype of this cat, that the reason why the colorization is one enzyme, and it gets turned on when the temperatures are low. Epistasis is another thing that Mendel would have never been able to truly understand. But what we're seeing here is that two different genes, one can control the other's outcome, and even though they're sorted through homozygous or homologous recombination and meiosis, sometimes a gene can actually affect another gene. And so in this case we have corn crossing, where we have two ears of corn that essentially were crossing to the F1 generation. And what strangely happens here is the corn here on the left is dominant for one of the genes, just signified here as having both uppercase A's, and recessive for the other gene, here signified by two lowercase b's. The other is the exact opposite. Recessive for A, dominant homozygous for B, and when we cross that, we get a heterozygous plant that has both the dominant and the recessive gene for both A and B. But in this case, it turns out A being a purple colored ear of corn. So what we see down here at the very bottom is what epistasis is showing us. Enzyme A, if it's available, meaning we either have to be heterozygous or dominant. If enzyme A is being produced, well, the color of the kernels is still going to be white. However, if enzyme B is available too, whether it be homozygous dominant or heterozygous, the plant's going to end up having purple kernels on it. So here, the only way B works is if A is available. And so here when we look at it, every single one of these purple ears of corn is going to have at least one copy of the dominant trait for both A and B. So here, obviously, here, yes, here, yes, 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 yes. You can go on through. Now, any of the white ears of corn is going to have a recessive copy of one of those genes, whether it be A or B. And when we see that, you can go through the other remainings, and you can see that that is in fact true that every single one of the white colored plants is going to have one copy of recessive genes for one of those particular enzymes, whether it be A or B. Now here's just a book that I find very interesting. It's written by a particular person that has both an MD and a PhD. And what they did was they started looking at different phenotypes that cause afflictions in humans, and trying to understand from a evolutionary standpoint why those particular phenotypes, or in this case genotypes, are passed on through different individuals. Why would something that's considered bad be passed on? And so, they look at the different histories of some of these particular situations and, go into maybe some possibilities of why these particular afflictions still exist. The ones that I like to look at are going to be alcohol dehydrogenase. This is an enzyme that's responsible for breaking down alcohol, whether it be from beer, wine, or hard liquors. And this goes all the way back to the ways that people used to purify water. People mainly of European descent purified their water by producing wines, beers, things like this that had alcohol in them. So therefore the enzyme alcohol dehydrogenase is incredibly important for breaking down these alcohols, because again, remember, this is before bottled water, before running water. The only way that you're purifying your water was to boil it, or filters would not have been a possibility at that time. So here, as part of the water purification process, people of European descent mainly focus on making wines and beers. People of Asian descent made tea. And so in this case, tea has no alcohol in it, and therefore there was no need for people of Asian descent to have the particular gene alcohol dehydrogenase. Today we can see this as different abilities of holding one's alcohol. Somebody that's of European descent may be able to go into a bar and have several beers and not actually feel like they're drunk. Some people of Asian descent, they have one small drink and they may be feeling or experiencing a lot of inebriation. And this just comes down to, do they have the enzyme to break down the alcohol they just ingested? There's some other examples here, but again, they're just all really kind of interesting from a standpoint of, I wonder where these particular afflictions came from. And so I've already spoken about the malaria affliction. This is an example here. You can basically take these two maps. The one on the left is people that have the sickle cell allele. And so, of course, it's a heat map, so green is going to be lesser amounts, light blue is more, and dark blue is very high rates. And what we can see here is right along the middle, or the belt of Africa, is Gabon and Congo. These are going to have some of the highest rates, especially even along the Gold Coast here. They're going to have high rates of mosquitoes. Malaria is carried by mosquitoes, and therefore those people would have needed to carry the sickle cell allele in order to survive past childhood to make it to reproduction age. Here we can see the heat map for where we find malaria within Africa, and if you lay the two on top of each other, they would pretty much coincide more or less. Here we talk about cystic fibrosis, and so this is another example that we've already spoken about, in the previous slides. All right, that's the end of chapter 12. I'm also going to put up chapter 14, so this will be the end of what's going to be on the next exam next week. You have both chapter 11, which I did give the lecture to after the exam last week. We have chapter 12, and we're going to have the first half of 14, which I will also be putting up.